All right, IB Bio students, this is what you've been waiting for. Topic 8, respiration and photosynthesis. So I love the Amoeba Sisters down here. Really cool videos if you're looking for something online on YouTube to help you with things. But yeah, photosynthesis, make it. Oh yeah, happy times for our glucose. And then cellular respiration. We're going to break you up and use you to make energy, ATP for the cell. So the essential idea is your body regulates all the reactions according to what your body needs. Your body does not want to make things that aren't necessary. Does not want to waste energy on stuff it doesn't need. So your body, when it needs stuff, it'll ramp up the production. When it doesn't need that stuff anymore, it'll shut down those enzymatic, metabolic pathways to conserve materials, conserve energy. Just like you should do in your own life. Remember, you shouldn't have stuff you don't need. And we'll get into this the second video, I believe, but this thing called bioinformatics, which is a real passion area for me, is basically digitized biological information. I don't know what that was. And when I talk about information, basically three things: DNA, which can also, what's going on there? And then proteins. And when we talk about all these things, we talk about sequences. So DNA, the sequence of A, T, C's, and G's, RNA, A, U, C's, and G's, and proteins. What's going on there? Proteins are talking about amino acid sequences. So remember, there's 20 different amino acids. So, which of those 20 in what order gives you that information. And using that database, they're able to see which genes are influenced by drugs or different chemicals, we might say. Influenced by, chemicals, aka drugs. So the drugs we take to stay healthy, based on your doctor's prescriptions, they can be tested by just using a database, which really speeds things along and allows for a lot quicker, a lot faster advancement in science. So enzyme catalyzed reactions, everything is based on enzymes in your body. And the first one we're going to talk about is glycolysis. So basically splitting sugar is what we're doing. We're taking a glucose molecule, we're adding phosphate. That's what phosphorylation means, and you'll see it then turns into this hexose biphosphate. Then it splits, which is what lysis means. So it splits into 2 3-carbon molecules called triose phosphate. It's then oxidized, and that means you lose electrons or hydrogens from that molecule, lost hydrogens or electrons, or it could mean you add an oxygen. In this case, we're losing these electrons and hydrogens. And that gives you ATP. And you might have noticed it takes ATP to start it, and you get ATP at the end. But remember, you add 2, you get 4. So that means you have 2 at the end. You have a net 2. You net out, say you spend $200 to do a party, you make $400. Ooh, that makes a cool $200. And another pathway we'll get into 8.2, not this one, or 8.3, is photosynthesis. And this right here, this is the Calvin cycle, or the light-independent reactions of photosynthesis. And that's an important metabolic cycle. You see CO2 from the air going in, ATP, NADPH, and then you get sugar at the end. As we were talking about, the amoeba cyst is making that sugar. Here we're breaking the sugar. Here we're breaking it. So break it. And over here we're making it. Man, my handwriting, I thought I was getting better with my graphics pad, but oh, it'll make it easier to see here. So here we're making it. This is the happy with glucose being happy, over here being sad to get broken up. All right. Enzymes, the main functional thing is they're lowering what's called activation energy. So you need energy to get started. Just like you need energy when you have, some kind of a sale or party. You can't throw a party without some buckos to get some drinks, some snacks, get the movie, whatever it is. The games, the decorations, the tablecloth. Tablecloth is very important. All the parties we had this year, students are being very insistent getting a nice tablecloth. Now I will say that really helps, really set the ambiance, the vibe of the party. Sorry about that. You don't have time to listen to that stuff. But what I have blocked off here, what do we call the beginning, the chemicals we start a reaction with? The reactants. Oh, that's some good handwriting. Kind of pat myself on the back. And what about what we get at the end? Everybody say it with me. Products. Sometimes I switch those around. Luckily, I did that time. And what we see two lines. This has a higher activation energy. Higher activation energy. This right here has lower activation energy. And this is what we see. This would be no enzyme. And this one down here would be with an enzyme. So you want to be with that enzyme. You don't want to put that much energy into things, right? And an enzyme is a type of catalyst. I did not mention that before. You will see that term. I kind of use them interchangeably. Enzyme is a biological catalyst. A non-biological catalyst is potassium iodide, and I used that the other day. Dropped it in some hydrogen peroxide and made mixed with soap, and I made some elephant's toothpaste. The potassium iodide increased, or it kind of drove that reaction, splitting the hydrogen peroxide into oxygen and water. And it wasn't used in the reaction, so that makes it, if it gets used up in the reaction, it is not a catalyst anymore. So some things that can affect our enzymes are competitive and non-competitive inhibitors. Now let's get into what the difference is. A competitive inhibitor will actually go into the active site and block, I'm sorry, got ahead of myself. This is normal reaction, where we have the substrate entering the active site. Entering the active site, this is the reaction happening. And at the end we have our products. And we'll see the enzyme, no change. It still is the way it is, still normal, hasn't been chemically altered, and it can be reused. Your body's natural recycling method right there. Now, ways that enzymes can be affected: you can have a molecule get stuck right in the active site, which means nothing's going to happen. It's blocked. The more competitive inhibitors you have, the slower that reaction will be. If you have enough of them, no reaction will happen, which is rare. Which I believe is a lot of poisons work. They block a lot of enzymes that can cause death. And we have a non-competitive inhibitor where you don't have it. See, it's kind of stuck on the side. It's not blocking the active site. So in most cases, the substrate can bind, but the reaction is blocked. So there's no reaction there. Sometimes the shape is changed. That interferes with the binding into the active site. Yeah, space. But you hear my words, active site, and therefore it kind of shifts the structure of the enzyme, and it doesn't quite bind the way it should. All right, that's it for part 2, or part 1.